Alright, so let's go ahead and take a look at a little bit further look at blood. We're going to look at plasma, clotting, and blood typing. Alright, so one of the elements of blood here is blood plasma. Plasma contains fibrinogen. Serum does not have fibrinogen. It also contains electrolytes like bicarbonate, magnesium, chloride, and potassium. It is straw colored and it is the liquid portion of the blood. It constitutes about 55% of the blood, and it is made of about 92% water. And the function is, of course, to transport those nutrients, gases, electrolytes, and vitamins to areas needed in the body. But it also helps to regulate fluid, electrolyte balance, and maintain pH. Alright, so by weight, plasma proteins compose the most abundant solute or dissolved substance in plasma. These guys stay in the blood because they're just way too large to pass through the membrane. So these are not normally used for energy sources. The proteins that we use for energy come from the digestive system, and then they're broken down. Plasma proteins hold water in the capillaries by osmotic pressure. We've probably talked about how water follows salt. Well, this kind of falls under that particular figure of speech. Water is going to follow proteins. Proteins help hold on to the water, all right? So we have three main types of proteins in our plasma: albumins, globulins, and fibrinogen. So these differ in composition and function. Albumins are the smallest, but they make up the largest percentage, right at 60% of proteins. These guys are made in the liver. These are most important for water balance, all right? If plasma protein concentrations are too low, we can have edema. The tissues are going to swell. Globulins are next, and we have three types. We've got alpha, beta, and gamma globulins. Globulins compose about 35 to 36% of plasma protein. These guys primarily transport lipids and fat-soluble vitamins, and they also, if we look at gamma globulins, serve as antibodies for the immune system, so that's very important. Fibrinogen, while only a very small percentage at 4%, is made in the liver, but it is very important for coagulation. All right, so coagulation is part of clotting, so this is a very important component of our vascular system. So blood gases and nutrients are also found in the blood. The most important gases that we have are oxygen and carbon dioxide. These are evaluated by using fresh blood from an arterial or artery. An arterial blood gas will be drawn, and that's going to determine oxygen, carbon dioxide, pH, and bicarbonate concentrations. And this can be used to diagnose and treat disorders of circulation, respiration, and even electrolyte imbalance. Plasma nutrients are going to be absorbed from the digestive tract. Normal levels of things like lipids can be biomarkers for metabolic, cardiovascular, or neurological disease. The nutrients are also going to include amino acids, simple sugars, nucleotides. So what are some of the other substances we might find in blood? We're going to have some non-protein nitrogen-containing or nitrogenous substances. So these contain nitrogen, but they're not necessarily protein. Amino acids, of course, come from digested protein. Things like urea and uric acid. These are byproducts of protein and nucleic acid catabolism. All right, and this composes about 50% of the non-protein substances. Creatine is part of creatine phosphate, which is in muscle tissues. And then we have creatinine, which is a product of creatine metabolism. So pay close attention to those words. So if you were to write these on your test, you need to be sure you know how to spell them and the difference. Creatine and then creatinine. All right. So you can have, we can check what we call blood urea nitrogen, and this can suggest kidney disorders if it is elevated. All right, because it indicates the health of the kidney. Kidneys excrete urea, so if they are not working correctly, they may have high levels of urea in there. We also have other electrolytes, and these are a small portion, but again, they're very important. Plasma contains a variety of ions, and we call these electrolytes. These are absorbed through the intestine or released as a byproduct of cellular metabolism. Sodium is a very important one. It is the most abundant along next to chlorine. Potassium is extremely important. Calcium, magnesium, chloride, bicarbonate, phosphate, sulfate, sorry. These are all important, some more important than others, and sometimes it just depends on what system you're looking at. One thing to really look at is bicarbonate. What is bicarbonate? Do you know what bicarbonate of soda is? That is baking soda. Baking soda can be used for a soured stomach, right? Because it can act as a buffer. We have a bicarbonate acid buffer system in our body, which helps to regulate our body's pH. All right, so bicarbonate is very important. Potassium is very important for heart health. If your potassium levels are off, your heart can actually stop. Calcium is important for muscle contraction, for instance. Chloride is related to, you have chloride leak channels where you can have certain respiratory illnesses if those don't function correctly. So these being off, you can see how that would be very problematic for you health-wise. So hemostasis is the stoppage of bleeding. Hemo means blood. Stasis means to halt or make stand still. So hemostasis, halting the stoppage of bleeding. This is the process that stops bleeding from damaged blood vessels, and they have several actions that can actually help to prevent blood loss, including blood vessel spasm, platelet plug formation, and blood coagulation. All right, so a vascular spasm can occur when cutting or breaking a small blood vessel happens. So blood loss needs to lessen almost immediately. It will spasm and slow down that blood loss. That gives us about 30 minutes to allow the platelet plug to then come in and form. The platelet plug is where these little cell fragments and platelets actually come in, and they stick here to the end. Look at that. They're sticking to the end of an injured blood vessel, which is basically any rough surface, which will happen to be the blood vessel. Especially they like to stick to collagen, which is in connective tissue, which is often what blood vessels are made out of. All right, so it's pretty handy. So blood coagulation actually occurs in what we call a coagulation cascade. Now, I had to memorize the entire coagulation cascade for biochemistry, but this is just basic anatomy, so we don't have to go in that great of a detail. But it is a cascade of events. This occurs when cellular damage happens and blood comes into contact with foreign surfaces. All right, a blood clot is going to form from those platelets. You're going to have a soluble fibrinogen that is going to change to insoluble fibrin. All right, and so that's going to help trap blood cells. You can see that fibrin and all right here, and that's the little orangish looking strings there. It's a hemostatic mechanism, stops the bleeding. All right, so this is going to cause the formation of a blood clot it's going to cause the formation of a blood clot by having a series of reactions. So you're going to have one thing that's going to activate the next in what we call a cascade, which is basically just like a series of dominoes falling. When one topples, it triggers the other to topple, all right? So we break this down into extrinsic or intrinsic, and this requires a lot of clotting fibers, and these guys have some names, and then some of them are just Roman numerals, and it's also very important to have vitamin K. So vitamin K is kind of an interesting thing here. That is a treatment here that can be used. I have little Pomeranians, which are about 5 to 10 pounds, and I had one. We live out in the country, so we had mice getting in, and so we had rat poison out. And what happened is rat poison interferes with blood clotting. And so over the period of time, what it actually does is deplete all the vitamin K so that you are not able to clot. And so over a period of time, the animals, after they eat too much of this, will bleed to death, usually internally. And so the treatment is just to give massive doses of vitamin K. So my little female Pomeranian, I got a hold of some, and we had to give her vitamin K for a period of about 2 weeks, which saved her life. So understanding these mechanisms is very important, because vitamin K is a very simple, easy to give medication. Or it's a vitamin, but it's a treatment in this case. This is a good summary here of what we were just talking about, the hemostatic mechanisms of blood vessel spasm, platelet plug formation, and coagulation. Alright, so we're going to go over this extrinsic clotting mechanism here. Remember, this is a chemical outside. Think exit, that means outside. So chemical outside of the blood is going to trigger blood coagulation. We have thromboplastin, which is also called factor 3, which is not normally found in the blood, that is going to be the toppling domino here. And then a number of events occur. And like I said, we're not going to go into the detail of this. If you move on to 3rd or 4th year and take biochemistry, you can memorize this whole thing. But we've got factor 6, 10, 5, 4, and 2. 2 is important. That is prothrombin here, okay? So you need to know some of these main words is what I want you to focus on. Prothrombin time is a blood test that we can actually use to evaluate how your clotting is occurring. So that's one you want to be familiar with. And so prothrombin, this is going to be triggered when blood contacts damaged blood vessels or tissues, all right? And so this is a positive feedback mechanism. So let's talk and remember positive versus negative feedback. Negative feedback does what? It returns something to a normal value. Well, positive feedback increases an event until a trigger stops the event. So remember, childbirth was always kind of our classic example of positive feedback. The contractions are increased by pressure against the cervix. The more pressure, the stronger the contractions get, and the cycle amps up until the childbirth actually occurs. Hormones are released, which then trigger the response to stop. All right, so the same thing is going to occur here. It is going to keep ramping up and continuing until there is some kind of trigger that says, okay, we've reached enough. All right. By the way, a thrombocyte is a blood platelet involved in the formation of blood clots. So the intrinsic clotting mechanism occurs inside, all right? So inside blood, and this is going to trigger coagulation. This is triggered by the Hegman factor, which is factor 12, all right, which is inside blood. Factor 8 is going to topple over or trigger 11, which then is going to activate 9, and that's going to join with 8 to activate factor 10, and this of course occurs when blood contacts a foreign surface. All right, and so I want you to just kind of know the main names. You don't need to know the order or all the factors necessarily explicitly from memory, but kind of understand the process. And so this top chart here is the one I would study the most. I would definitely go through this one down here and look at the clotting factors, okay? But what I really would like you to focus on is this one right up here, okay? You need to know thromboplastin, prothrombin, fibrinogen to fibrin, okay? So just those right there, I want you to really learn these steps in that top table there, 14.3. Do look through 14.9, understand kind of the clotting factors, but you really should know this top chart that we just went through. So here is an example of the cascade, the intrinsic and the extrinsic, and how they both lead to factor 10. All right. Once you get to this stage, they act the same way. So know that the difference is extrinsic is outside the blood and intrinsic is inside the blood. Other than that, they're fairly similar. This one here obviously has a few more steps to it, and it gets more complex down here. Fibrinogen and fibrin are very important components, as well as thrombin and prothrombin. It's hard to say those, they're tongue twisters. In these guys here, these are kind of like precursor molecules. So what will happen is something will be tacked on, or usually something's taken off to make the next molecule, which activates it, all right? That last slide, I must have been talking whenever it started. That has a really funny pose for me on there. So I know sometimes that looks kind of funny. I try to sit still and look straight at the camera, but I'm not a still kind of person, so it catches me in some kind of humorous looks at the beginning of these. So I apologize for that, but you are free to laugh at them. That's fine. All right, so what is the fate of blood clots? After a blood clot forms, it's going to pull away from the edges of the blood vessel and pull itself back while squeezing fluid from the clot, right? And so there's a growth factor that stimulates the smooth muscle cells and the fibroblasts to come in and repair damaged blood vessel walls. Plasmin here is what actually digests the blood clot, so we break it down. An important thing here to note clinically is going to be the difference between a thrombus and an embolus. You've probably heard of an embolism, but they're very similar, and again, it's location. So embol means stopper. So an embolism is a mass lodging in and obstructing a blood vessel, okay? A thrombus is just an abnormal blood clot. So basically, a thrombus is a blood clot, and you can see that right here. And then here is an embolus. It is a blood clot that is moving. It's on the move. All right. So embolus, think energy. I don't know. It's on the move. All right. So that's the key difference there, is whether or not it's moving. All right. And so that does make it particularly problematic if it is moving and it goes to someplace more dangerous. That's when an embolism becomes scary, is if it goes to the lung or the heart or something. If it's down in your leg and you have a clot in your leg or in your arm, you don't want it to move to your lungs, right? You don't want to move it to your heart. All right, so we don't want our blood to clot all the time. So how do we inhibit clot formation? That is important, and we actually can use this medically when we need to prevent coagulation. So you've got a smooth lining in your blood vessels, which gives little place for those platelets to stick to, right? They stick to rough surfaces. So the fact that our blood vessels are smooth is one thing that helps to inhibit blood clots. As a clot forms, fibrin is absorbed or absorbs thrombin, and that can help prevent the clotting reaction from spreading on throughout the body, because you do not want to throw blood clots all over your body. If blood clots are thrown all over your body, that is basically what we call DIC, or disseminated intravascular coagulation, which is a very, very serious condition, and that's where you have blood clots basically everywhere, so we don't want that to happen. Antithrombin is a factor that will inactivate thrombin by binding to it and blocking it from participating with fibrinogen, which then goes on to trigger fibrin. So we also have cells like basophils and mast cells that secrete heparin. And we actually can give heparin intravenously as well. So heparin is an anticoagulant. So remember, we have those mast cells. They release heparin and they release histamine. All right, so this is one of my favorite components of the lecture, blood groups. Blood group typing. I always find this really interesting. I like drawing this one out on the board and putting the little antibodies, and even though my drawing skills are not that great on the board, I don't know, for some reason I really like doing it. So in about the early 1900s, we were able to identify the ABO blood antigen gene that gave us blood type incompatibilities. Blood type incompatibilities were actually one of the reasons way back then you had to get a blood test to get married to make sure that you were compatible, and particularly because of the Rh factor, which we will get into. So today we know there's about 31 different genes that are known to contribute to surface proteins, which are the features on red blood cells that determine whether or not you are compatible with another blood type. Understanding the difference between an antigen and an antibody is important, because we talk about them a lot. We talk about them a lot, especially when we get to microbiology. Antibodies, of course, are self cells. They are cells that our body makes. So this is a protein, basically, that is going to react against a very specific antigen. And we have antibodies in our body that are sitting ready and waiting to react to almost any antigen that could possibly come around. It's really quite amazing. So an antigen is basically anything that stimulates cells to produce antibodies. This can be a foreign invader, like a bacteria that's going to have antigens or a virus on the outside of their cell, or it can be something like pollen, all right? And so antigens don't necessarily have to be living. Of course, viruses aren't living anyway. Another term to look at is agglutination. A gluten means glue together. And so this is the process of gluing together. So this is that clumping of blood cells in response to a reaction between an antibody and an antigen. And so when they are not compatible, they will agglutinate or clump together and cause problems. So the ABO blood groups are based on two of the major antigens that we find on red blood cell membranes, antigen A and antigen B. And we're going to look at that in detail. That is what determines whether or not you have the antibodies. Alright, so here this is a really nice diagram. If you have blood type A, you can see here you have A antigens, alright, on the outside of your cell. So your body will recognize those. And so you don't have antigen B out on there. Antigen B, you can see, is a different shape. And so your body will not recognize antigen B, and you will have antibodies to B. So you cannot receive, if you are type A, you cannot receive type B because the antigens and antibodies don't match. All right? Now, if you are type B, we have the opposite situation. You can see the B antigen down here, okay, and it's all around here, but we don't have any of the A antigens. And so we have antibodies against the A antigens, so we can't receive anything that is type A. We can receive things that have type B or that have nothing, but we can't receive things with type A. Now, if you are type AB, you have both antigens, all right? So you can accept blood with A or B because you don't have antibodies to either one, all right? And so, let's move on and then we'll get to that in a moment. All right, and then so type O doesn't have those antigens at all. So type O can be given to A because it doesn't have any antigens to respond to. It doesn't have any B antigens. It can also be given to B because it doesn't have any A antigens, and it can be given to AB because it has no antigens, right? So that's what makes O the universal blood donor. The problem there is if you are the universal blood donor, you can't receive from anybody else, okay? Because you can give to everyone because you don't have the A or B antigens. But because you don't have the A or B antigens, you have both antibodies. So you cannot receive neither A nor B. So while you can give to anyone, you cannot receive from anyone other than O, okay? AB is exactly the opposite. AB is the universal blood recipient because it can receive O because there's no antigens. It can receive B because it has B. And it can receive A because it's got A antigens, all right? So it can receive from anyone, but it can't give to anyone because it's got both antigens, and everybody's going to respond to one of the antigens, right? So a person with type A blood can donate to another person with type A, right? A person with type AB can receive from somebody with AB or O. So type O is the universal blood donor, but can only receive O. AB can receive from anyone. Now we're not looking at positive and negative yet here. AB can receive from anyone, but can only donate to other AB. All right. And so we also have another factor we're going to look at, and that is the RH factor. RH factor is another protein that we found outside of the blood, and this is what determines whether you are positive or negative. And that just means you either have the RH factor antigens or you don't, okay? This was actually, RH comes from the name from rhesus monkey because this is how they discovered it. So if you are RH positive, you have the antigen for the Rh factor. So if you have the antigen, you will not make the antibodies. About 85% of the population is Rh positive. If you are Rh negative, you do not have the antigen, so you will make the antibodies against the Rh positive type. This is problematic for women that are childbearing, okay? So if a man is positive and the mother is negative, the mother has a chance of having a negative baby or a positive baby. This is normally not a problem during the first childbirth because there is no mixing of blood. It's very rare. It can occur, but it's very, very rare for there to be a mixing of blood until birth occurs. What happens when childbirth occurs, though, is you develop those antibodies. And once those antibodies are in your system, and you can see these in this picture here, after the birth of the first child, once those antibodies are in your system, there's nothing we can do, okay? Those antibodies will then go in and attack the blood of the second fetus, okay, which can be very, very bad. You can have a condition called erythroblastosis fetalis, or hemolytic disease, all right? And so we have thankfully come up with a way. I know my grandmother was negative, and I am Rh negative. And when I became pregnant with my first child, my grandmother was very, very concerned because in her time there was nothing they could do to prevent this. And so typically the first baby would be fine, but after you had one child, the child could have some really severe problems, or they could even perish, okay? So it was very concerning, but we came up with something called RhoGAM, okay? RhoGAM. RhoGAM is an immune globulin, and it basically prevents the mother from developing those antibodies. And so what they do during pregnancy, during the 5th month, they will give one shot of RhoGAM then. It's human immunoglobulin. And then after the child is born, if the child is positive, they will get another shot of RhoGAM within 72 hours. If the child is negative, it is not necessary. So this is yet another reason why prenatal care is so very, very important. Because like I said, once you develop these antibodies, there's really nothing we have been able to do to fix that situation.